Started?
No, it does.
Yes.
Now it has?
Yeah.
Oh, okay, sweet. Hi, I am Miss Sloan, and this is my honors bio class, and we are reviewing chapter 25, control of gene expression. So if you're wanting to jump in the Pear Deck right now, LPBMQD. Right? So I made a list of the topics you want to be comfortable with. So if you kind of look through a few of these, what are the jobs and roles of binding sites like the promoter and the operator, the genes, regulator, and structural, and then enzymes and molecules like RNA polymerase, repressor, and lactase? Do you know how these work together? Number two, can you differentiate, describe the 5 levels of control in eukaryotic gene expression? And then number three is, all things cancer and the characteristics of cancer cells, the 2 key genes, the difference between benign and malignant, what is angiogenesis, the causes of cancer and the treatment of cancer, and then have you done your highly suggested reading and thinking? Okay, so that you know where you are on that. I'm just going to start working my way through it. So the first thing we need to get over is that every cell in our body, sorry, I need to switch the lights. Right. Every cell in our body has the exact same DNA in it, right? And we have what's called differential gene expression, where some genes are turned on and others are left off, or they're turned on for different times. And so to learn a little bit more about this, just a second, I need to fix this. All I can see is myself. I need to see the Pear Deck. To understand this a little bit better, we start by talking about prokaryotes. So we go with the simplest first. All right. So prokaryotes are going to adjust, making whatever proteins they need in the moment. Okay. So when we look at a prokaryotic cell, remember it has a single circular chromosome that when it boom, 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 when it makes mRNA, as soon as it starts building the mRNA, it can immediately be translated. Why can't that happen in a eukaryotic cell? Why don't we see that happening in a eukaryotic cell? Because of not the cell membrane, but the nuclear envelope, right? So where transcription takes place is inside the nucleus. Where the ribosomes are is outside of the nucleus, right? So transcription and translation are separated in 2 different regions of the cell. But since a prokaryotic cell doesn't have a nuclear envelope, as soon as you're making that mRNA, it can immediately start to get translated because it doesn't cut out any what? Introns. Yeah, what it has, it expresses. You all right with that so far? Okay. So then we looked at, here we go. We looked at some different regions on the DNA itself. So I want to walk through those first. The first thing I want to talk to you about is a regulator gene. And the regulator gene will code for a repressor. In the case that I'm talking to you about that we use as an example, it's an active repressor, which means when this DNA is transcribed into mRNA and translated into a protein, this protein is good to go. It starts repressing. How does it repress? This repressor will bind onto a region called the operator. The operator doesn't code for anything. It's just a binding site for the repressor. But when the repressor binds on to that operator, it spills out over into another region as well. What's the other region it spills out onto? The promoter. And who wants to bind to the promoter? Yes, RNA polymerase binds to the promoter to begin transcription. So it cannot transcribe these structural genes, whatever those are coding for. It can't transcribe those or get them translated because the repressor is blocking its ability to bind. So that's the first premise we need to get and understand. If you were going to draw on this slide, I would know regulator gene is a binding site, I take that back, erase. Regulator gene codes for a protein, a repressor, but the promoter is a binding site, the operator is a binding site, and the structural genes, in our example that I give you, those structural genes, if they were transcribed and translated, they would code for enzymes that digest lactose. Okay, so that's the premise. Let's see how it gets changed. Okay, so here in this case, here you can see the regulator gene. We've got an active repressor. It's binding to the operator. As such, RNA polymerase cannot bind to the promoter. These structural genes are turned off in essence. It's like the light switch is off. But right now, you're not home, so it's okay if your bedroom light is off because you don't need it right now, right? These genes are not needed right now. These genes code for an enzyme to digest lactose, and currently there is no lactose present. Currently, you are not in your bedroom. You do not need the light on. Okay? If lactose does become present, there we go. If lactose is present, lactose will bind to the repressor, and now the repressor lets go of the operator. So now RNA polymerase can bind to the promoter. So you walk into the room, now is a good time for the lights to go on. So RNA polymerase binds the promoter, boom, 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 make your mRNA. Your mRNA is translated. Every 3 bases is a codon complementary to a, and you're putting amino acids together to build a protein. This protein will digest lactose. All the lactose in the cell, including the one binding to this repressor to turn that, to get that repressor away. As soon as that lactose has been digested, then we can go back and the repressor will bind back on there. But that's okay because I don't need these genes anymore anyway. Okay? Ask the person next to you, do you get this? Are you feeling okay? Do you want to ask any questions about that? Okay, anything you want to ask me? Because see, what I want you to be able to do is for it to make sense to you, because then you don't have to what it, if it makes sense. Memorize it, right? So you need to know what everybody's job is. What's the job of the regulator gene? Yeah, to code for a repressor. What's the job of RNA polymerase? Yeah, it wants to bind to the promoter and transcribe the structural genes, right? That's its job. Yeah. What is the job of the operator? It's the binding place for an active repressor, right? So and what's the job of the structural genes? In our example, they code for what? Something to digest lactose. Okay. So if it just kind of makes sense to you, then you don't have to spend any time memorizing. Okay. All right. Then we level up and we talk about control, levels of control in eukaryotic cells. Eukaryotic cells have a nuclear envelope. 3 of the controls occur inside the nucleus, 2 outside of the nucleus. So the first level of control is your DNA even available, right? What do we call it when the DNA is tightly condensed, wound, and put away? What do you call it? Heterochromatin. Okay, it's condensed and put away, it's heterochromatin. When it's available, it is called euchromatin. Good. Okay, then we say, okay, let's say it is available. Let's say it is euchromatin, right? Transcriptional control, the most critical one, RNA polymerase, what? Binds to the promoter. Can RNA polymerase bind to that promoter? Does it need any assistance? Is there a repressor that is getting in the way that would inhibit that process? Sweetheart, I'm in the middle of recording a review. Did you need something? Today?
Yeah.
Sweetheart, I did not have a phone given to me. I'm so sorry. Do you have a friend who can call your phone and see? I probably won't ring because you have it on silent, right?
Yeah.
Can you do Find My iPhone or anything like that? Oh, okay. I'm sorry. Did you check in the office?
I'm gonna give one of my friends got it a bunch
Okay, but check in the office too, okay?
All right.
All right, sweetie. Good luck. Okay, so transcriptional control, can RNA polymerase bind to the promoter? Can it get help? Does it need help binding to the promoter in order to make your mRNA? Post-transcriptional control, now I've boomed. I have my mRNA. So what's this one about? Putting on a cap and cutting out before it leaves the So if anything prevents, facilitates, doesn't let it get out of the nucleus, that is post-transcriptional control. Translational control, can the mRNA go out and get on a ribosome, right? Are the tRNAs free to land on that mRNA? Can you build your protein? Post-translational control is, now I have my protein, my primary structure. I might need to what it? Fold it, right? Get it in the right shape for it to do its job. So how I ask about that is I make a question to see if you can understand which level of control it is. Like I might say, you've taken a poison that inhibits mRNA from leaving the nucleus. What level of control is it interfering with? And you would tell me post-transcriptional. Because I didn't stop you from making the mRNA, but the mRNA couldn't get out of the nucleus, and that's part of cap tail, cut out the introns before it leaves the nucleus. Does that make sense? If I said something is interfering with you taking it and putting a protein from its secondary structure to its tertiary structure, you would tell me that was post-translational control. Something is blocking mRNA from getting on the ribosome, translational control. The DNA is permanently as heterochromatin, chromatin structure. Okay, do you see how that works? So I just need you to be able to recognize that. So then I gave you some different pictures about that, right? We talked about whether DNA is tightly coiled or not. I grabbed this from an AP. This looks new to you, right? This is just the process of how DNA coils. You don't have to worry so much about that. Okay. The way DNA coils, I really thought I took out this slide too. I grabbed this from, but on this part, do you recognize the central dogma over here? DNA codes for RNA, which codes for protein. So epigenetics is anything above the genes where it can influence the way that DNA is condensed or decondensed. Do you remember when we talked about, I'll get it to this slide, you will remember. Do you remember when we talked about bar bodies? Whether an X is a bar body or not, right, is going to be whether or not you have the genes available on that particular X chromosome. We talked about it with calico cats and why the calico cats where you see the different colors, would that be a female cat or a male cat where you'd see the different coloring? Female, because you don't know which X is active, right? Whereas a male cat, they only have one X. Good. And about different colors of eyes, we talked about that as well. Okay, this one right here, do you remember when we talked about transvesomes, jumping genes, right? So that can influence what DNA is available if it is jumping around. Okay, then next, we talked about transcriptional control. That's weird. Did it just go back to that or was it ever showing? Please don't seize. I should not have said the words, right? I should not have said the words. Hang on a second. All right, let's do this. Pause. Pause. Is it playing? Okay, we're playing. Good. Sorry, we had some technical difficulties, but we're back. All right. So post-transcriptional transcriptional control is, does RNA polymerase bind to the promoter, right? Post-transcriptional control, we're putting on a cap or a tail or cutting out the introns before it leaves the nucleus. Perfect. Okay. And then when we, let's see right here, you can see exon recombination. Do you remember when we talked about cutting out the different exons, right? Or sorry, cutting out the different introns and leaving the exons, because the exons are the ones that get expressed, right? Because it doesn't sound like the way it is. So just keep remembering exons are expressed. Okay, I'm hopefully going to go to another page. We'll see. Okay, and then translational control. Can mRNA get on the ribosome? Can you translate that into a protein? And then post-translational control. okay, is any folding pattern that you impose upon those proteins after you have formed them. Are you okay with that part? Okay. So now, moving on, hopefully. So we're going to talk about cancer. And then first, we talked about whether a tumor was benign or malignant. Benign, it's staying put, right? It's encapsulated, but a benign tumor can become a malignant tumor and it can spread. Now, when it spreads, it has to be able to, emerge. It has to get that mutation. But one of the things that usually keeps it in check is that it doesn't have enough nutrients in order to expand and grow. So angiogenesis is that process, and I do have a slide for that, but angiogenesis, do you remember what that is called? When the blood vessels grow towards it, right? And if it gets all the nutrients it needs and its waste taken away, then you can have even more cells. The longer it's there unchecked, then it might gain that mutation in order to spread, to metastasize. And if you remember the two places we didn't want it to go, what are the two places we don't want it to go? Yeah, the lymphatic system and the circulatory system, because that basically gives it a path through your entire body. Okay? And then we talked about two genes. We talked about the protooncogenes and the tumor suppressor genes. Now, protooncogenes, you have about 100 different protooncogenes. In and of themselves, they're not bad. They're just in a position of control. So if you have a gene that's in power and it mutates, then it can abuse that control. And what they do is they promote the cell cycle. And if you recall, I was saying like it's a army general who controls a lot of troops. And if that army general goes bad, then all the troops end up obeying bad orders. The other analogy we gave is that oncogenes, which proto-oncogenes can mutate into oncogenes, we said it's like your car, it's like stepping on the gas, right? Which in and of itself, it's fine if you have a problem with your gas, like it's stuck, as long as what's still working, your brakes. And that's what tumor suppressor genes are, is they're coding for your brakes to put a stop to it. Okay, so those are the two big gene categories that can end up having cancer too, that control for the proteins that you end up having cancer. Those cancer cells look markedly different when we look at the characteristics of them. Okay, normal cell, and we looked at the different characteristics. You remember we kind of ping-ponged back and forth with a cancer cell. You have small cytoplasm, lots of nuclei, and it's very coarse when you look inside the nucleus, so you can visually see the difference. And then with these differences, they don't follow the rules. And one of those rules was anchorage dependence, where it has to stay put, connected to another cell. If they lose anchorage dependence, that can help them metastasize. And the other one was what? Normal cells exhibit what? Contact inhibition, right? Which means I can tell we're crowded, so I'm going to stop growing. That's contact inhibition. Cancer cells don't do that. They just keep going, and going. And then if these cells lost anchorage dependence, they could move to other parts of your body as long as they had access to those tissues, right? Okay. So then somebody just colored out those pictures. They did not like it. Angiogenesis, you remember, was when blood vessels grow and it can resupply that neoplasm, that group of cells, that benign tumor, and give it the oxygen and the nutrients it needs to spread. Okay, then the causes of cancer, we talked about heredity, right? We talked about different genes. We talked about like breast cancer genes and things like that. So you can inherit it from your family. And then we said there were things in the environment, like not living a healthy lifestyle and smoking and getting exposed to UV radiation. Those types of things can cause cancer. When you look at the diagnosis of cancer, we talked about mammograms and pap smears, and you can get your blood checked for cancers. You can have genetic tests to see if you're more likely to have cancer. These can be confirmed with biopsies or several imaging techniques. You do not need to memorize each of these imaging techniques. Just know that there are imaging techniques that you can see if you have a tumor or not. And then the treatments, the typical treatments are things like surgery to have it removed, or radiation, or chemotherapy. Maybe in the future we will have cancer vaccines, but we already have one, right? HPV, right? If you don't get that virus, that prevents you from some cancers. P53 therapy, which is helping you with your gene that codes for your brakes, and then drugs to prevent blood vessels from growing towards it. That would be helpful to prevent the spread of cancer. Okay, and then that was it. Did you have questions? You're good? All right, good job, you guys.